Revolutionary technology that can edit genetic mistakes is getting attention and scrutiny this morning. CRISPR could help rid us of diseases like cystic fibrosis, muscular dystrophy, and even HIV and cancer. Think about that. But many scientists, including CRISPR's developer, Jennifer Doudna, are calling for a moratorium on its use in humans. Tomorrow, she holds a global summit on the ethical issues. And only on CBS This Morning, Doudna shows us why, for all its promise, CRISPR is surrounded by controversy. And what is CRISPR?
CRISPR, first of all, it's an acronym. It stands for Clustered Regularly Interspaced Short Palindromic Repeats, a huge mouthful. You can see why we use the acronym CRISPR.
So I'm sorry, what's CRISPR again? Geneticist Jennifer Doudna gets asked that question a lot. A researcher and professor at UC Berkeley, Doudna has become a spokesperson for a gene editing technology she's credited with developing. That mouthful known as CRISPR. I've heard it compared to essentially like a film editor, slicing a bit of film.
I would say that's a great analogy, yeah.
How does that work then?
Well, think about a film strip, and you see a particular segment of the film that you want to replace. And if you had a film splicer, you would go in and literally cut it out and piece it back together, maybe with a new clip. Imagine being able to do that in the genetic code, the code of life. You could go in and snip out a piece and replace it with something that corrects a mutation that would cause disease.
That's incredible.
It's incredible.
CRISPR has generated immense excitement because it's fast, cheap, and can cut and paste genetic code with great precision. It used to take months or years to alter a single gene. Now, that can be done in a matter of days. Could it end cancer?
What I'm excited about there is the potential to use the CRISPR technology to program a patient's immune system to recognize tumor cells in a precise way.
Could it cure, at some point, virtually any disease?
I don't know about any disease, but I think any disease that has a genetic basis is something that could be treated using the CRISPR technology.
And imagine, Doudna says we could expect to see clinical applications of CRISPR within the next few years.
This is no longer science fiction.
But alongside CRISPR's promise come some fears of its perils, like embryo editing that could lead to designer babies. What is the dark side of this technology?
One of them is, of course, making changes to human embryos which become permanent. So we're talking about something that would affect human evolution.
You could have an instance where a lab is creating lots of human embryos just for the sake of experimenting on genome editing on them, right?
If you're asking me, could that be done technically? The answer is it could, right? Could it be done, with current regulations in place? Certainly not in the U.S. No,
or Europe.
Right? Or Europe, right. Yeah.
There's still a lot of other countries other than the U.S.
Well, this is the thing, right?
Yeah,
Science is global, right?
Yeah.
Science is global. And, there are different cultural viewpoints on that kind of application.
In April, Chinese scientists reported using CRISPR to edit the disease genomes of human embryos for the first time. The experiment was a failure, but it sparked concerns worldwide.
I and my colleagues have called for a global pause.
Doudna has long been vocal about the need to set ethical boundaries and is convening an international summit tomorrow in Washington, D.C. What do you hope comes out of that?
I think it would be great if we can at least get on the table the key issues. It's hard to imagine that there would be a consensus by all of the parties at the table about how to proceed, but I do think that the first step is really to have that kind of open conversation.
Now, scientists are bringing attention to a new advanced reproductive technology. It's called in vitro gametogenesis, or IVG. And with this process, adult cells, like skin cells, can be reprogrammed to behave like embryonic stem cells. And these can then be stimulated to grow into eggs or sperm in a culture dish. And these so-called artificial gametes are then used to form an embryo for implantation into an adult womb. Last year, Japanese scientists revealed the birth of mice from eggs made from parents' skin cells. Now, in theory, the need for healthy human sperm and eggs could one day be bypassed altogether. IVG could replace IVF as a treatment for infertility. George Staley is the dean of the Harvard Medical School. He is a pioneer in stem cell research, and he and his colleagues published a paper recently about some of the ethical and legal questions raised by this new technology. And I asked him when this might become a reality in humans.
Well, it's already a reality in mice. And we expect that there's no real biological distinctions between the ability to generate gametes in a dish, whether it's mice, monkeys, or humans. Now, it hasn't worked in humans yet, but we anticipate that sometime in the future it will. And so my colleagues and I have decided to sound the alarm, to raise the issues, both scientific and ethical, so that we can have a public debate and decide the permissible and impermissible applications of this technology. There's many ways that this new technology is going to teach us about human development, about reproduction and fertility, and may give us new ways of treating infertility. So the positive medical applications are tremendous and very exciting.
As a scientist, I can tell that you're very excited about this upcoming breakthrough, about what could happen next. You also express that you're conscious and aware of the ethical implications of this. So have you been able to game out in your mind sort of the unintended negative consequences of this technology and how to avoid that?
Well, if we think back 40 years, this debate also started around the time of the birth of baby Louise Brown, the world's first test tube baby, born from in vitro fertilization. What was anticipated at that time, and concerns were raised, was that human reproduction might become commodified, that we would have really large-scale reproduction in a dish. We also raised concern at that time, and once again, for coupling gene editing technologies in a way that would allow us to create human enhancements. Now, with advances in gene editing technologies, especially a very feasible one called CRISPR, we now have a remarkable capability of going in and editing genes. Now, of course, this could be done once again to eradicate disease, but what is, I think, more concerning for individuals is whether or not we would use gene editing for human enhancement. It could start with something as simple as influencing your offspring's height or eye color. But what if we learn to be able to manipulate things like learning and memory? Then it becomes more of a concern as to whether the technology will be used only by the privileged few or be available more globally.
You know, scientists today have access to some pretty powerful technologies. In this era, though, among the public, there is a rising, rather, lack of trust in experts, a lack of trust in scientists. Do you fear of a public backlash to this kind of research?
Well, I certainly hope not. I feel very strongly that science is one of the last bastions of truth-telling. Our goal is really to be transparent, open, and candid about both the medical opportunities and the ethical challenges. I think by scientists coming forward and engaging actively with ethicists and members of the public, we will win the kind of confidence that we need to be sure that these new technologies are used for human good.
Science is one of the last bastions of truth-telling. That was part of my conversation with George Staley, dean of the Harvard Medical School. Now, Staley and his colleagues raise the prospect of same-sex couples being able to have biological children, or a person infertile, say, from cancer treatments, also having a chance at biological children.
A baby comes into this world unlike any other baby has before. For the first time, doctors in Ukraine used a new type of three-parent IVF technique so that a previously infertile couple could have a child. To clarify, this isn't the first child born with DNA of three parents, but she was the first to be conceived using a technique called pronuclear transfer. In this instance, doctors in Kiev combined the father's sperm with the mother's egg and then placed those genes into an egg that was donated by another woman. Then they implanted that egg in the mother's womb, and on January 5th, she gave birth naturally to a baby girl. The baby has her parents' genetic identity with the trace amount of DNA from the donor woman. But her conception is controversial. For one, the science of three-parent IVF is new and still highly experimental. It also raises a number of ethical concerns and fears over the possibility of a future with so-called designer babies.
This is it. Human stem cells being injected into a pig's embryo. For the first time, scientists are seeing how our cells grow and interact inside an animal. This pig, the first known chimera.
The ultimate goal for this type of research is to whether we can use this technology to generate human tissue and organs. That's going to give us a lot of unique insights.
Right now, scientists are nowhere near that goal, but recent studies have shown how that process can work using gene editing. Scientists would delete a specific piece of an animal's DNA, so its embryo wouldn't be able to grow a heart, for example. Then they'd inject that embryo with human stem cells, which move into the void and grow a human heart. The embryo would then be put back inside the animal to develop.
The possibility of growing human cells and tissues inside animal is an exciting first step for the dream of generating tissues and organs for transplantation into humans. We're still far away.
To date, everything the researchers have done is legal, but what they're doing is highly controversial. The embryo is only being allowed to develop for 28 days. It's not allowed to be born. Bioethicists say this research raises a lot of uneasy questions.
The boundaries between what it means to be human and what it means to be non-human can become quite blurred. Also, it's not great for the animals. There's risks for them. There's no benefits for them. It may help a lot of people, but the moral status associated with this is a deep, deep, deep question.
That word, chimera, from Greek mythology is meant to elicit fear. But the scientists argue they are trying to solve a problem and save lives, not create a monster.
The woolly mammoth is a cultural favorite, a long-extinct animal that can still capture the imagination. We commonly accept the woolly mammoth became extinct at the end of the last ice age 10,000 years ago due to a combination of climate change and human hunters. But suddenly, this loss might not be permanent. There are a handful of projects across the globe working to bring extinct animals back from the dead. They call it de-extinction.
What I would emphasize in de-extinction is de-extinction of DNA, not so much of species.
Leading one of those projects is Dr. George Church, a professor of genetics at Harvard University. He's a genius.
This is the tRNA that I solved helped solve as a teenager.
To create new mammoths, Dr. Church started with a close relative. Then, he made some key changes to its genome.
Our lab and others have developed a whole variety of genome engineering tools, and one of them is CRISPR. The way that CRISPR works is that you calculate in the computer a particular set of 20 A's, G's, and T's in a row, and then the CRISPR protein cuts, and then that cut alters the gene.
Proponents of de-extinction believe that bringing back the mammoth would represent a turning point for what we can do with genomes, and the positive effect our work with genomes can have on the world around us. And of course, it would give us the chance to say hello to an old friend.
The surgery worked safely as intended.
On YouTube, He Jiankui claimed he's done something no other scientist in the world has.
Two beautiful little Chinese girls named Lulu and Lala came crying into the world as healthy as any other babies a few weeks ago.
Twin girls, he says, from an embryo that was genetically edited, part of an experiment aimed at protecting children from ever contracting HIV. During IVF, a technology called CRISPR was used on embryos, disabling a particular gene that allows HIV to enter a cell. But the approach, restricted in the US and much of Europe, drew an international outcry.
There has been consensus that to take an embryo with altered genes and put it in a woman to create a child is just beyond the pale in terms of what's morally acceptable.
And that's what he says he's done.
That was exactly what he has said he's done.
The Chinese university where He was on leave said today he never reported his research to the school, as questions swirl about whether the procedure fully worked and whether editing the baby's genetic makeup could have unintended consequences, perhaps making them more susceptible to other diseases.
This is a slippery slope to designer babies. That if I start altering the genes of a child, I could alter other genes of a child.
He has released little evidence to back his claims.
I believe family need this technology, and I am willing to take the criticism for them.
A brave new world the world may not be ready for yet.
I can't wait to have a baby. Now, I know what you're all thinking. She's just a kid, but I've got it all figured out. First, I'll attend my dream college, then start my dream job. Of course, marry my dream husband, and together we'll design our dream baby. I've always wanted a baby with blue eyes and dark hair, maybe a little bit of astigmatism because babies with glasses are cute. Really, by the time I'm gonna be able to have children, I'll even be able to decide the height, the intelligence, and any desired trait. This sounds like a dream, doesn't it? Being able to design your perfect child. Actually, being able to design generations and generations. But who is giving consent to this? Certainly not the unborn baby. I mean, who are we to alter millions of years of evolution just for a baby with blue eyes, a high IQ, and world-class speed? We have the technology to play God, to mess with nature. It's called CRISPR. What a funny name. With CRISPR, we can cut out undesired parts of DNA and replace them with arbitrarily generated desirable traits. Does it still sound like a dream? Picture this. In 15 years, you and your partner have a normal child, which means that she is not genetically modified. Let's say her name is Maya. Your neighbors, on the other hand, decided to go down a different path and named their genetically modified daughter Emma. Now, Emma is smarter, prettier, more talented, and scientifically proven to be better than your child. She's the perfect child, perfectly designed, but there's always been something off about her. She has to go to a different school, lead a different life which is very different from that of your child's. I mean, after all, she's a designer baby, perhaps a trophy child. Emma lives just next door, and somehow you know that she never gets sick, she never gets hurt or trips easily. She's definitely an asset to the government, who will most likely never have to spend large sums of money for her health care. Well then, maybe even the government might start to promote the production of designer babies, and your little Maya will find herself alone, filled in a world with perfect humans. It wouldn't be the first time that the government controlled or tried to control childbirth, whether the number, the mental, or the physical qualities of the offspring. A modern example could be China's one-child policy. But I think that the most frightening example of eugenics was during the Nazi period. Hitler wanted a superior race, the Aryan race. He went through great lengths to achieve this, firstly by concentration camps, and then by sterilizing or executing individuals which he judged to be handicapped or cretins. Is this the world that you want your children to inherit? Is this what you want our race to become? Oh, and Maya, you'll love her just as much with her messy hair, slightly asymmetrical nose, and that little cough which never seems to go away. But maybe, just maybe, you won't be able to stop yourself from comparing her to her friend Emma. And trust me when I say that you won't be the only one. So will colleges, employers, potential mates. And what if one day she comes back crying, asking, why am I not like Emma? All you know is that society has changed. Between your child and Emma, there's an abyss. A superior race has been born, and your child does not belong to it. It's time to ask ourselves really important questions like, who are we? What will we become, and who will decide? IVF is a modern example of designer babies. Selective breeding, which is a type of baby designing, has been around for thousands of years. IVF is already an accepted way of reproducing for infertile parents or those who run the risk of passing on dangerous genetic diseases. In other words, it's already a method of bypassing natural evolution. Now, imagine being a parent who decided to reproduce using IVF. And the doctor mentions that for an extra $1,000, he can eliminate the risk for your child of ever getting Alzheimer's. Now, I'm pretty sure that each and every one of you would accept his offer, because as a parent, everyone wants the best for his or her child, whether the best job, the best education, or the best genetic trait. Parents are already influencing their child's life so much by imposing a religion, an education, sometimes even a hobby. So why not give them the best traits they need to thrive in the society they live in? In other words, a virtual guarantee of success. Now, this reality seems far away, almost as if it was a post-apocalyptic movie plot. But it's actually closer than what you might imagine. Experimenters in China have already tested this technology on human embryos. Now, it did not go too well, but these are only the early stages of the trial. The good news is that we have an excuse to avoid the production of designer babies. They are simply too dangerous. But what about when this is not an excuse anymore? The first child born from IVF came to light a little over 40 years ago. And since then, 5 million children have been born through that technology. 5 million in 4 decades. That's a lot. Now, a child like Emma may not be too far away, and the choices scientists make will define and maybe tear apart society as we know it. But then again, where do you draw the line between saving a life and creating a trophy child? And most importantly, who will draw that line? Think about your own children for a second. Is there anything you would change about us, except that we're always asking you for money? And if that answer is yes, do you still want a perfect child? Now about the baby, I can wait. Thank you.
A Chinese researcher has created an international controversy over science and ethics after claiming he helped make the world's first genetically edited babies. According to the Associated Press, the researcher altered embryos for 7 couples during fertility treatments. One resulted in the birth of twin girls. The claims have not been independently verified. They also were not published in a medical journal where they could be vetted by experts. Now, this kind of gene editing is banned in the United States. The changes can be passed to future generations and could harm other genes. Our Dr. David Agus is an oncologist and leads the USC Westside Cancer Center and is here to talk more about this. A lot of people in the medical community are buzzing about this. Can you tell us how this doctor was able to do this?
I mean, this is wild. This is an enormous step. And what the doctor did was he took sperm out of a man and took it out of the semen and put it together with an egg. This is in vitro fertilization. And then when he made an embryo, he put in this thing called CRISPR that's able to surgical precision change one of the 3 billion letters of the DNA code. And he changed the code for the HIV receptor, for the AIDS receptor in the cells. And then that embryo was put into a woman.
So is this process able to eliminate HIV because it's being promoted that way?
Well, in this study, when you edit this receptor, yes, you can eliminate HIV. But in these 2 twins, only one of them had it fully taken out. The other one had it partially taken out. But at the same time, it raises the risk of death of flu and increases the rate of getting West Nile virus. So it's not a perfect treatment. And the problem is there's a lot of off-target effects. This enzyme is promiscuous. Even though it could change one letter, it happens to maybe change other things in the DNA.
So we don't know any of the unintended consequences yet. Obviously, this raises a lot of ethical questions. This is banned, this process, here in the United States. Do you ever see this actually being possible amongst U.S. doctors?
Well, to me, this is an experiment on children, right? These were parents. The fathers actually had HIV themselves, and the mothers did not. And then they consented them. On the consent, it said HIV vaccine. It didn't say gene editing experiment. This is something where we can get rid of inborn errors of genetic disease potentially, but a lot more science needs to be done. It is way too premature to put it in kids because we're not going to know the answer for 10, 20, 30 years whether it works.
But this is a subject we clearly will be talking about going forward. We've been talking about CRISPR for the past 2 years. What can you tell us about this doctor?
Well, this is a doctor who trained in the United States and then went to Europe. You know, there are CRISPR studies ongoing now in the United States, But we do that in adults to treat a disease. This isn't passed generation to generation. Once you hit what we call the germline, the DNA of a child, it then is passed to the next generation, the next. So these changes are permanent.
Huge changes in the world of science. We'll be hearing more from this doctor as he speaks at a conference in Hong Kong. But great to hear you this morning talk about it.
Today we brought you what? We brought you the story of how new genetically altered fruits and vegetables and even animals could soon be entering the marketplace. But now ask yourself something. Should we be changing a plant or animal's genetics? And if so, to what end? What about restrictions? Some? None? Then there's the question of whether we are or should play God, right? Joining us now is Paul Thompson. He is a professor of agriculture and ethics at Michigan State University. So he's, man, talk about the perfect person to have on this segment. Let's start with you explaining for us as easily as you can for our viewers who are not scientists what this gene editing thing really is. What are we actually doing when we say gene editing?
Well, we've been modifying the genetics of plants and animals in some sense for centuries, but fairly actively through breeding programs for many decades. And over the last 30 years, we've developed techniques that allow us to do that with more precision, targeting specific traits that are based in the genes. And gene editing is the latest trend in some of that work that's been going on. It allows us to make even more precise changes that target specific spots in the genes. So in some respects, it's a continuation of what we've been doing, but with more specificity.
Is there a limit to what we should do when it comes to this particular field? I, as a layman, would think, oh, this sounds a little mysterious, almost scary, but you as a scientist would say what?
Well, I certainly think that there are reasons to be concerned about the safety, and I think that there have been quite a few safeguards that have been put in place that are going to focus on the safety of actually consuming these products. I think one of the issues that's particularly significant when we start to talk about animals is that unlike genetic engineering of a corn plant or a soybean, the animals have feelings, and so we have some additional ethical concerns that have to do with the way that a genetic modification affects the way that an animal feels, the way that they fare or do in the world.
What is the worst case scenario? What is the doomsday scenario, if there is one, with our continuing this approach?
Well, I'm not sure that there's a doomsday scenario. You know, there are cases, even with just ordinary plant breeding, where people have failed to pick something up, and you know, you wind up with cases of toxicities getting into the food system. The ordinary potato is toxic in the green parts of the plants, and even with just ordinary breeding, you can transfer some of that toxicity to the plant. So the key there is having some of the safeguards in place. I don't actually think that genetic modification itself is tied to you know, sort of end of the world type scenarios. But we could do things that we probably shouldn't do, and probably would alter certainly our sense of our place in the world and with respect to some of the plants and animals that we eat.
Shortly after Japan announced that researchers received government support to create mouse-human chimeras, a Spanish team of scientists have produced monkey-human chimeras. The research was conducted in China to avoid legal issues. The team wants to develop chimeras, organisms composed of cells from 2 or more species, capable of growing human organs. Similar experiments using pig or sheep embryos have faced technical challenges, likely because the animals are genetically distant from humans. Primates, being more closely related to people, may offer more promise. So far, the human primate embryos have only been allowed to develop for a few weeks at a time before organs have formed, according to Estrella Núñez, a biologist and administrator at the Catholic University of Murcia. In no case is the gestation brought to full term, she says in an interview with El País. I don't think it is particularly concerning in terms of the ethics, because you are not taking them far enough to have a nervous system or develop in any way, it's just really a ball of cells, says Robin Lovell-Badge, a developmental biologist from the Francis Crick Institute in London, in an interview with The Guardian. But, if you allow these animals to go all the way through and be born, if you have a big contribution to the central nervous system from the human cells, then that obviously becomes a concern. The real question we raise here at the Atlantis Report is, how much DNA do you need to change before a living thing is no longer classified as human and loses basic human rights? By basic I mean the right to their life without being euthanized for organs. Why is experimenting on chimeras bad but experimenting on monkeys not bad? What makes humans so special that a few drops of our DNA makes a monkey untouchable? Monkeys are already highly intelligent, sentient, not fully sapient like us beings. They experience emotion, fear, and pain just like we do and have highly evolved brains. If say monkeys share 99% of DNA with humans and you create a chimera human monkey or monkey human, how close to 100% do you need to get before euthanization and murder? There is a danger here of the creation of a subhuman class which is essentially human but without human rights.
A controversial world first. Spanish scientists say they've managed to create a human-monkey hybrid embryo. They claim they're looking for a way to grow transplantable organs for humans. Seems they took a monkey embryo and deleted genes responsible for creating organs. They were then replaced with human stem cells which could be transformed into any kind of tissue and organ. The experiment was aborted after 2 weeks, but if completed, the scientists claim human organs would have grown inside the monkey, potentially transplantable then into a human body. We put the ethics of all that up for debate.
Look, a monkey is not a human, and therefore humanity doesn't necessarily apply. Obviously, using animals to create human organs is cruel, but then again, I'm much more concerned about the loss of human life than I am about the loss of animal life. Now, in an ideal world, we wouldn't harm animals in this way, but I do think that if you're being given a choice between the death of a child and the death of a monkey, I think we do have to accept the death of the monkey.
The problem is, Andre, that when it comes to organ transplant, that's difficult enough when the organ is, for example, a heart or a kidney that's being moved from one human being to another. In this instance, what we're talking about is moving an organ from an animal to a human. I'm not sure that I would like Cheetah the monkey to be the source of my donated organ. I'm not sure about you.
I think, David, in that situation, there is a level of desperation. So obviously, having any transplant is something that is not ideal. It's not something that's desirable, and obviously, it's extremely dangerous. But I think one of the problems we have at the moment is the number of people who die simply because no organs are available. Organ transplants, which are a relatively new technology, don't necessarily go to plan. People have to take a lot of drugs to suppress their immune system so they don't reject the organs, and that can lead to very serious problems, including cancer. If we can make organ transplants safer and better, that will be a positive step forward.
The road to hell is paved with good intentions, and just because those ushering us along that road wear white coats does not mean that it's a destination that we should be going. This is scientifically and morally flawed, and it's something that we really shouldn't embrace.
Obviously, what we don't want is to cause animals suffering, but at the same time, we have to prioritise humans first, simply because I think it's such an important thing and it's such an important area of medicine and science that we have to tolerate a level of suffering.
I think the idea of inflicting such hurt on them will repel many people and think that it's completely inappropriate. I don't think the animal kingdom is there for us to harvest at will. I think it can be done, certainly in this area, things can be done in other ways that don't involve harvesting organs from within living animals.